When we grow the bacteria in a growth medium where sufficient nutrient supply is available, so what happened? Bacteria grow well and develop, survive, reproduce, produce their biomolecules. Biosynthesis is going on. Cell is going to divide. It means replication, cell division, every aspect of growth, they are covered here, where the all all the eminent condition available there. Now, organism rely on regulatory network to integrate and process signals from various sources and to orchestrate the transcription of gene controlling a range of cellular process. The bacteria or microorganism, they relay the regulatory system. So all the regulation is going properly. Biosynthetic mechanism is continue. It means the regulatory relay system that allow to synthesize the food material, synthesis of biomolecule, synthesis of nucleic acid, synthesis of amino acid or protein, everything is going on. So there is a signal is released. How this signal is released? When cell having enough quantity of energy and raw material inside. So these all the signal they relay and they continue the growth activity. Now uh, Sunita Singh entered now today, but Twinkle Viskarma is not there. Anyway, so organism relay their regulatory network. For why? For biosynthesis purpose. It is on right way regulation where all the eminent condition is available. So integrate and process signal from various source to orchestrate the transcription of genes controlling a range of cellular process. So a range of process are there, physiological, biochemical process are there. These all coordinatingly conducted by the cell due to the relay of this regulatory signal. When the signal is relayed to close or shut down this process, so they are going to shut down. Why they shut down? Condition is going to adverse. When condition is adverse, so suppose the starvation is appear, food material is not available, carbon source is not available, or temperature is increased, means temperature is stress, osmolarity is stress, or other stress, they are appear in the cell or in the through the surrounding. So cell relay different type of signal and they manage another set of genes. The continuous growth process set, that is continuously. But another one set is start. That set is very, very important and that close the some system and some new system they are appear. Suppose it in a cell, there is a damage. Okay, damage is big. Then immediate there is a release of signal. These signal, they in the form of suppose it SOS signal. SOS signals are released. So what happened there? Their immediate large number of new gene or new set of gene, they start to transcribe, they start to work. Some they shut down the metabolic process, biosynthesis is completely halt. Now cell division is halt due to the lone system, lone A. Lone A gene when appear, it stop the cell activity, so cell division is shut down. So due to this anaerobic aerobic shift, there is also a basic problem because surrounding condition or the environment in which the microorganism is survived, that condition is changed. So there is a need of protect the bacteria, protect the organism itself, and stop some unnecessary or unwanted process or mechanism where energy is waste or that process is not appropriate. So this type of regulatory mechanism, there is a great regulatory network that integrate the process of signal, and these signals they control, they are known as the regulon. Regulon is the signal that regulate a module. Module means a gene. So gene is transcribed or not. So it regulate by the regulon. So first of all, alarmon is there. Alarm बजा और उस अलार्म का सिग्नल किसने सुना? जो उसकी मैकेनिज्म में पार्टिसिपेट करता है वो है रेगुलॉन, और रेगुलॉन ने तुरंत अपना रेगुलेटरी सिस्टम को पॉजिटिवली नेगेटिवली मीन सप्रेस करना या उसको बूस्ट करना, ये दोनों काम रेगुलॉन के हैं. तो दे डू देयर वर्क. So lot of regulon they release, they shut down the various process and new process they start. This is a common system where signaling system is concerned. So now in this chapter we discuss the organism relay a regulatory network to integrate the process of signal from various sources and to orchestrate together uniform एक साथ सारे एक्टिविटी चलने लगे. Transcription of gene controlling a range of cellular process. In the range of cellular process, the controlling of transcription. Transcription में आपने देखा कि ट्रांसक्रिप्शन एक जीन का होना है जिसका प्रोडक्ट काम करना है, मगर उसको ट्रांसक्राइब करने के लिए और भी कई सेगमेंट हैं जीन के. कौन कौन से सेगमेंट हो सकते हैं? एक तो जीन और एक उसका कंट्रोलिंग सिस्टम, ऑपरेटर, प्रमोटर, एनहेंसर, बूस्टर, जहां अलग अलग फैक्टर आके अटैच होते हैं. ये सारे कंट्रोलिंग साइट्स हैं जीन के ऊपर. इसके बाद उसका एक रिप्रेसर, और रिप्रेसर को भी कंट्रोल करने का एक और रेगुलॉन. तो ऐसे कई रेगुलॉन. इट मींस the function of gene when expressed. So there is a number of gene they involve to express their function. सिर्फ एक अकेली जीन लैकोपेरोन में आपने देखा ZYA, तीन एक मल्टीसिस्ट्रोनिक जीन है, मल्टीटोनिक जीन है. इस जीन को एक्टिवेट करना, इसका फंक्शन करना, इसके प्रोडक्ट को बनाना या ट्रांसक्रिप्शन करने के लिए ऑपरेटर रीजन, प्रमोटर रीजन, स्टार्ट पॉइंट और सीजी बॉक्स आपने सुना होगा, -10 सीक्वेंस, -35 सीक्वेंस, -55 सीक्वेंस और ट्रांसक्रिप्शन फैक्टर सीक्वेंस. सो एंसिलरी प्रोटीन अटैचमेंट साइट्स. तो यहाँ ढेर सारी ऐसी साइट बनी है. इसका मतलब और दूसरी जो साइड है उनसे ट्रांसक्राइब होके प्रोटीन आएंगी, यहाँ अटैच हो जाएंगी और वो अपना फंक्शन करेंगी. They may positively regulator, they may be negatively regulator. So it means the regulatory network is there that stop the gene function or that boost the gene function. It is very, very important a relationship in the signaling regulatory network. Importantly, changes in their architecture of this regulatory network play a significant role in survival or adaptation of organism to changing environmental condition. When changing environmental condition, they are appear. So what is the function of regulatory network? They cope with this changing environment condition and they take decision how we modified the functioning of the cell. They stop the activity means negative control. They boost the activity means positive control. So overall, they involve in the regulation. So in this way, importantly, changes in the architecture of the regulatory network play a significant role in survival or adaptation of organism in changing environmental conditions. Okay. So this is very, very important part as far as introductory system is concerned. The FNR in your course, okay, definitely you got short notes over on the FNR. What is the full form of FNR? Fumarate nitrate reduction. FNR protein. So fumarate nitrate reduction protein play a central role in the global oxygen response of variety of the bacteria. Number of bacteria like eubacteria, archaebacteria, or thermobacteria. This FNR protein that play a central role in the global control of response of variety in the bacteria. In Escherichia coli, it is most studied bacteria. The fumarate nitrate reduction gene is the master transcriptional regulator. FNR is the master regulator in the Escherichia coli of the transcription. It can regulate transcription. It means master means so many genes can regulated by the single product, that is FNR protein. So in Escherichia coli, FNR is the master transcriptional regulator of the transcription between aerobic and anaerobic shift. What is the meaning of shift? The growth, aerobic growth, anaerobic growth. When aerobic growth is very good in the Escherichia coli, sudden we remove the oxygen. So what happened there? The bacteria that face change. There is a environmental condition is change, aerobic to anaerobic. So there is a aerobic to anaerobic shift occur in the bacterial physiology and biochemistry. So this is known as the shifting or transition between the aerobic and anaerobic shift. The regulation of FNR is achieved by cycling the molecule between three states in the process depend on oxygen. So oxygen presence, it stimulates the FNR. FNR going to activate and then it do their function and regulate the global regulation of aerobic to anaerobic shift. It control. So FNR is considered as a global oxygen response or global oxygen controlling regulator or transcriptional regulator. Okay. Now, as a facultative anaerobe, when Escherichia coli, it can survive in oxygen-containing environment and in an oxygenic environment where oxygen is not present. So it, it is considered as obligate aerobe or facultative aerobe. It means facultative means it cannot survive in the presence of oxygen, but the compromising facultative aerobe, it means it can also survive in aerobic and anaerobic condition. Obligate are very strict. Obligate anaerobe or obligate aerobe, they are strict. But facultative, they are fluctuating. They can survive in both the condition. So as a facultative anaerobe, Escherichia coli regulate respiration based on the availability of electron acceptor. How many type of electron acceptor are available? So electron acceptor, you know, in the electron transport chain, where we call back our knowledge. You focus your attention on the redox coupling, where respiratory chain is arranged. Suppose it NADH2 is a molecule that enter into the respiratory chain. What happened there? So in the respiratory chain, there is a number of molecule, electron transporter, electron carrier, electron acceptor, terminal electron acceptor. They arrange in a series. Okay. So NADH2, this molecule, it is highly reduced. Its redox potential is 0.034 kilovolt. So it can reach on that side where NADH2 actually reach on that side where the specific type of molecule, those accept the electron and proton from this highly reduced nicotinamide dinucleotide molecule. So here iron sulfur containing protein, okay, ferridoxin is there, pheophytin is there. So ferridoxin, suppose it can take the charge, it's also the redox potential is 0.034 kilovolt, and NADH2 is also 0.03 volt. So both having a similar redox potential. So when NADH2 reach to the negatively charged minus 0.03 volt potential, so their situation is ferredoxin is available that receive this molecule, take the reducing power, NAD oxidized form is removed. Now ferredoxin is reduced. Ferredoxin now shift their reducing power to the iron sulfur protein. Then iron sulfur protein shift their uh, reducing power to cytochrome C3. Then cytochrome C3 shift their reducing power to cytochrome C1, and cytochrome C1 shift their reducing power to the nickel cobalt containing cytochrome system B1, B2, B3. And finally, the A1, A3 complex, it is known as peroxidase. It, there is a oxygen is available. So this molecule pick up the oxygen on one side, and another side, it pick up the electron and proton and convert the oxygen into the water. So this is known as terminal electron acceptor. So in most of the electron transport chain, oxygen is terminal electron acceptor. So similarly, here in Escherichia coli electron transport system, there is also a oxygen considered as, or oxygen is located as the terminal electron acceptor. अभी जो हमने ये सिचुएशन आपको समझाई, इलेक्ट्रॉन ट्रांसपोर्टेशन, हाउ द इलेक्ट्रॉन कैन ट्रांसपोर्ट, ओके, in the redox couplers, in between the redox coupler. So different bacteria like Bacillus subtilis, Escherichia coli, Pseudomonas, Enterobacter aerogens, or Azotobacter, or Gymomonas mobilis. These all the bacteria having different type of electron transport chain, and their cytochrome system is different. Iron, sulfur, nickel, sulfur, copper containing protein, cobalt containing protein, they are also different. And the molecule that is reduced and that donate their electron to this molecule in the electron transport chain, so they are also different. Suppose it FADH2, its redox potential is 0.029 kilovolt. So on the other hand, the NADH2 having NADH2 and NADPH2, they have high reducing power, 0. minus 0.034 kilovolt. So it means different molecules, those containing electron, or they are able to donate the electron, they have different potency, different potential of electric charge. So they reach on the different side. But in most of the cases, electron transport chain, there is a terminal electron acceptor. Sometimes this terminal electron acceptor may be iron, sometimes it is magnesium, sometimes inorganic compound, or sometimes organic compound like sulfate or ferrous ion, ferric ion. So they are also accept the electron. They also situate. So we can say the different kind of bacteria, iron bacteria, sulfur bacteria, manganese bacteria, hydrogen bacteria. So different type of bacteria. Sometimes carbon is also used as terminal electron acceptor. So methonogens में बिल्कुल अलग प्रॉब्लम है. तो इसका मतलब माइक्रोबायोलॉजी में there is a vast range of terminal electron acceptor. So here in Escherichia coli regulates respiration based on the availability of electron acceptor molecule. Oxygen is the preferred acceptor. It is very common electron acceptor considered as terminal electron acceptor, and it repress all other type of respiration and fermentation. It can repress if oxygen is not available who receive the electron. So there is a different type of shift immediate. The condition is changed, oxygen is not available, it means terminal electron acceptor is not there. So bacteria release their series of regulon or signals. These signals, they shut down or repressed the other type of mechanism like respiration and fermentation mechanism. they inhibit respiration aerobically or fermentation anaerobically. That direction is changed. In the absence of oxygen, nitrate become the favored oxidizing agent. When oxygen is not available, then most of the bacteria, including Escherichia coli, they favor the nitrate as a terminal electron acceptor, is it? So it is oxidizing agent. Four global regulator are there: FNR, fumarate, nitrate, reductase, protein. ARCA, NARL, and NARP control the various pathway of respiration. Which one pathway is conduct? Where EMP pathway is going on, ED pathway is going on, glyoxylate pathways are going on, HMP is there, pentose phosphate pathway is there, or glycolate pathways there, or other pathways they may be run over there. What types of mechanism is there, or sulfur or iron, what types of pathway they run. So number of other molecule, oxidizing molecule, including FNR protein, ARCA protein, NARL protein, and NARP control the various pathway of respiration. They receive the electron, so they are going to inactivate. Or whether they receive the electron, they are going to activate. Once they activate, they initiate a series of signals, and these signals, they liberate the series of regulon, and they regulate the activity of modulon like operon. What is the modulon? There, operon is a modulon where the activity is modulate, means it may be repressed or it may be expressed fully. Is it? So this is the basic mechanism of various regulatory system where the aerobic and anaerobic shift is going on. Now, FNR, fumarate nitrate reductase protein, take its name from the mutant deficient in fumarate and nitrate reduction. This name is coming from the mutant. Those are negative. FNR negative means they are where the FNR protein gene is mutated. It is damaged. Okay. So its role is to sense oxygen. First of all, when oxygen is sensed by such type of organism, when they grow in the oxygenated environment, they survive well because terminal electron acceptor is there. But oxygen is available, so their growth shift is not occur. Aerobic to anaerobic shift is not occur. So why? Because they have a sensatory system. This must be sensed the deficiency of molecule like oxygen. So oxygen is not available now. So how we conduct the mechanism? Oxygenated respiration or oxygenic respiration is not possible. So this type of microorganism they sense. Now time is warning, last 5 minutes, within 5 minute time is over, है ना, बिना बताए टाइम खत्म हो जाएगा। तो जब तक जितना हो जाता है उतना चला लें। कि आपसे कुछ पूछ लें? इतनी जितनी बात हुई है इस पार्ट को तो पूरा कर ही दे। Its role is to sense oxygen to determine whether the cell will grow aerobically or anaerobically. The regulation of this global transcription factor, which is the focus of this study, involve a cyclic network of interaction at the post-transcriptional level and auto-repression at the transcriptional level. So you understood or you already learned various type of regulation. Regulation during the transcription, regulation by repressor, regulation by transcriptional factor, or the regulation by various protein, those are attached in the structural gene or their location with structural gene. And after some time or another type of control mechanism, post-transcriptional, when transcription is going on, how this regulation, row dependent, row independent. Then transcription is complete. So after transcription, translation is going on or initiate the transcription. So laureate in case of galactose operon. We already discussed arabinose operon, where the CRP protein and cyclic AMP, they are the catabolic activator and catabolic repressor protein involved in the controlling of operon. Operon must be fully expressed or little expressed or close. So in the presence of glucose, in the absence of glucose, in the presence of galactose, in the presence of arabinose, what types of mechanism is going on? So due to the activity of catabolic activator protein and catabolic repressor protein along with the activity of cyclic AMP. So CAP binding protein, CAP, that is very important, that play important role in regulation of that operon. So here, similarly, its role is to sense oxygen. So this FNR, negative strain, how they sense. So from there, this system is discovered, and FNR is a system that sense oxygen and determine whether the cell will grow aerobically or anaerobically. So FNR is sensitive for oxygen. When oxygen is there, it is sensed and aerobic respiration is promoted. If oxygen is not there, it is sensed and anaerobic mechanism boost. इसका मतलब दोनों तरफ का सेंसेशन इसको मिल रहा है. ऑक्सीजन है तो एरोबिक रेस्पिरेशन चलना चाहिए, पाथवे उस तरफ के ऑन होंगे, और यदि ऑक्सीजन नहीं है तो वो शट डाउन हो जाएंगे, एनारोबिक पाथवे चलेंगे. इसका मतलब एक मॉलिक्यूल दोनों सेंसेशन में अलग अलग एक्टिविटी करेगा, राइट. यदि ऑक्सीजन है तो एरोबिक पाथवे चलेंगे, यदि ऑक्सीजन नहीं तो एनारोबिक पाथवे चलेंगे. इसका मतलब मॉलिक्यूल की एक्टिविटी, it is totally depend upon the presence of oxygen or not, absence of oxygen. In the presence of oxygen, this molecule when bind with oxygen, it can operate another type of system or regulate regulons. And when oxygen is not attached, it can recognize the another type of set of regulon. It means anaerobic respiration may be conducted. So have different mechanism, different environmental condition, response by bacteria. So the regulation of this global transcription factor, which is the focus of this study, involves a cyclic network of interaction at the post-transcriptional level and auto-repression at the transcriptional level. Okay, then we will discuss later. अगले पार्ट में इसको आगे डिस्कस करेंगे. ओके, यदि आपके पास कोई अभी ऐसी प्रॉब्लम हो जो आप नहीं समझ पा रहे हैं तो यू कैन टेल मी. कोई कुछ पूछना चाह रहा है? 6 लोग, 7 लोग हो गए थे, एक कोई कम हो गया है ना. कौन चला गया? सुनीता सिंह विड्रॉ हो गई, उनका नेटवर्क का प्रॉब्लम है ना. सुनीता सिंह नहीं है. अनीशा देखो टाइम पे आ जाती है. अनीशा समझ में आ रहा है आपको? अनीशा, कैन यू हियर? हाँ, आज जो कुछ भी बात हुई ये समझ में आया? हाँ, ये वाला पार्ट एडवांस पार्ट है मॉलिक्यूलर बायोलॉजी का, कि हम माइक्रोबियल फिजियोलॉजी में मॉलिक्यूलर बायोलॉजी की जीन से रेगुलेट कर रहे हैं. तो इट इज नोन एज बायोफिजिक्स. और बायोफिजिक्स में फिजिकल फैक्टर और केमिकल फैक्टर दोनों इन्वॉल्व हैं. और जीन को तो हम छोड़ ही नहीं सकते कि फिजिकल और एनवायरमेंटल फैक्टर्स को सेंस कौन करेगा? वो हैं जीन्स. और जीन्स जब तक नहीं आएंगे ये माइक्रोबियल फिजियोलॉजी कंप्लीट नहीं होगा. तो यहाँ आपने अभी माइक्रोबियल फिजियोलॉजी में देखा कि जीन्स जो हमारे सेल में है, एक जीन की एक्टिविटी कंट्रोल करने के लिए उसके कंट्रोलिंग रीजन है, रेगुलेटरी नेटवर्क है. ये रेगुलेटरी नेटवर्क एक सिग्नल रिलीज करते हैं और वो सिग्नल डिसाइड करते हैं कि कौन सी जीन को अब खुलना है और कौन सी जीन को बंद होना है. विच वन जीन should be express their function and which one gene should be shut down and repress their function. So this is very, very important mechanism where the gene, they play, the same gene play in two different situations are there, and one gene is there and one gene product that play different role in different environmental condition, like oxygen is present, so that do different activity. When oxygen is absent, so that gene do different type of activity. So this is known as the global regulatory network. Is it? ये बात आपको अच्छे से समझ में आना चाहिए. तो सबको समझ में आ रहा है? ट्विंकल विश्वकर्मा सुनाई पड़ रहा है?
यस सर.
हाँ, आप हमेशा लेट होते हैं, आपको 9 बजने का पता नहीं चलता. हाँ, थोड़ा सा घर में बात,
चलता है सर.
हाँ नहीं, घर में थोड़ा सा मैनेज करके रखिए. 1 साल और बचा है आपकी एमएससी को, सेकंड सेमेस्टर देके इतने में और लोग कोऑपरेट कर दें आपको तो आपका काम बन जाएगा, है ना? हाँ, ओके,
यस सर.
गो अहेड, है ना? You also convey my wishes and sentiments because I'm worried about my student. है ना? They must listen me. Once they listen me, एक बार यदि आप हमको सुन लें तो ये पार्ट आपको सरल लगने लगेगा, ऐसा मुझे विश्वास है ना? कुछ सरल लगने लगता है.
यस सर.
हाँ,
यस सर.
तो एक बार आप सुन लें, क्योंकि इससे जो अटैच टर्मिनोलॉजी है आप उसी में गोल गोल घूमते रहते हैं, आपको नहीं समझ में आता है, वही हम एक्सप्लेन करते हैं. तो उससे क्या है, इस सब्जेक्ट में क्या होना है, क्या हो रहा है, इसका एक फ्रेम आपके दिमाग में तैयार होता है. तो टीचर को सुनना, है ना? टीचर इज नॉट नाउ बिकम एज ए गुरु, ना, टीचर इज बिकम एज ट्रांसपॉन्डर, विच वन इज राइट एंड हाउ टू स्टडी. ये सब चीजें हमको बताना, इसलिए हम टीचरों की जरूरत होती है. तो आज जो इसमें इलेक्ट्रॉनिक्स के जमाने में जहां हमारे पास नेट है और सारी नॉलेज वहां अवेलेबल है, वहां टीचर का इंपॉर्टेंस उस नॉलेज को समझने में क्लियरिटी लाना. दिस इज आवर डायरेक्शन. तो ये एक इम्पोर्टेंट बात है ना? आई एम वरीड अबाउट द सुनीता सिंह और बिंदु और गुलाबी. ये थोड़ा सा हमारे कमजोर विद्यार्थी होते जा रहे हैं क्योंकि ये काफी दिनों से क्लास नहीं अटेंड कर पा रहे हैं. अब क्या किया जाए? तो ये जो सिचुएशन आज आपने पढ़ी, इट इज वेरी वेरी इंपॉर्टेंट पार्ट, वन मिनट वी हैव रिमेन. फिर भी किसी के पास यदि कोई प्रश्न आपके पास हो तो यू कैन स्पीक. जल्दी बोलो, आप कोई प्रश्न है तो? कोई कुछ कहना चाहेगा? कोई कुछ बोलेगा? हाँ, निकिता दास, क्या आप कुछ कहना चाह रही हो? कहाँ है निकिता दास, दिख रही है कहीं?
नो
हाँ है.
सर.
हाँ, बोलिए, कुछ पूछना है? जब टाइम रहता तभी पूछ लिया करो.
नो सर.
ओके, सब समझ में आ गया. आरती शर्मा, आपको समझ में आ रहा है?
यस सर.